Going live. And
I'm
we're
not on
live.
Minecraft.
Hello, everyone. Welcome back to the best show on air. Today, we have a fun episode, and we're just going to get started, right? Yeah, so make sure to...
We have Mike here.
Yep. We have
Hello.
Mike. Hey.
Thanks, Mike.
You're welcome.
So make sure that if you have any comments to leave them in the chat below so that...
You gotta add the Gravity Max squad.
Yeah, exactly. So yeah, just leave any comments in the chat below so that we can respond to them, that'll be fun. So yeah, I guess we can just
let's hop
get
right
started.
into it.
Yeah, sure. Okay. So, this video is not intended for children under 13 years old. If you are under 13 years old, please do not watch this video, as the topics we discuss are meant for those with at least a high school level of education. If you are not 13, you will not understand the scientific topics discussed in this video. Okay. So, also visit the Gravity Max website at googlesites.com. It's a great website. Yeah, just check it out. Also, visit Gravity Max on social media, Facebook, Instagram, and Twitter. We have links for everything in the description, as always. Also, follow Sebastian on social media and check out his YouTube channel, 128 Seconds. He has some great videos. They've been on our channel, but I think, next Wednesday is probably going to be the last one that we put on the channel, so you'll have to go there.
Yeah.
So, So I'll stop freeloading, So no more free content. Now I'll be doing something else.
Oh, it's still free. It's just on my channel.
Yeah. Well, no more free content for me.
Oh, I see.
Now I have to do something every other Wednesday. But yeah. So links in the description. Check out his channel. Great channel. So those videos are going to become exclusive to his channel pretty soon, by the end of March. So yeah. Okay, also, A Truly Dead Rock is available on Amazon.
Best book, New York Times bestseller.
Yep, yeah. And link in the description. Sci-fi book, fun book. I'm already halfway through the sequel. I'm on the second draft. And it's going to be great. So, yeah. All right. Genetics. This is our topic.
Genetics.
Yeah. This is our topic for today. We're going to be looking at the processes within the nuclei of our cells, how our DNA works, how our RNA works, protein synthesis, all the different features on the cellular level. So it's kind of biology, also kind of chemistry. It's a mixture. So that's what we're going to be looking at.
Yay.
Yeah, genetics is everything. Everything has genes, Yeah. I'm talking everything's got chromosomes. There's that one plant that has 1,000 chromosomes. Humans, we only got 23 pairs,
Yeah.
I'm saying genes is everything.
It's funny. For some reason, a literal, the plant, a potato, has more chromosomes than a human being. And it's what do they need all that space for? What are they coding?
Something.
Exactly.
Yeah. So the animal with the largest number of chromosomes is the hermit crab, which has 127 pairs or 254 chromosomes.
That's crazy. Yeah, I know.
But the plant.
Yeah. I know dogs have something 30 pairs.
It's the ferns. Yeah, ferns have over 12,000 or 1,200.
Oh wow. So it's interesting how humans, even though we talked last week about the brain, even though we have the most complicated brain, we don't necessarily have the most complicated genetic code. So genes are weird. We haven't fully figured them out yet, but we are working on it. So yeah, let's start off with base pairs. So DNA is essentially just a code written in base 4. So base 4 gives you 4 digits, 0, 1, 2, and 3. And DNA codes all of the information about you in this base 4 sequence. So instead of binary like a computer 0101, it's 01234. So these are your 0, 1, 2, 3, and 4. So instead of digits, it's just 3 or 4 different molecules, and then a 5th one comes into play, but it's weird. So let's start off with adenine. So adenine is, yeah, you see DNA sequences are often represented.
Where did you get these great graphics from? But
oh, I made them.
Tell us about them.
I made these graphics.
Oh, you
made
them. You made them.
Yeah, I made them.
He's an artist.
Yep, thank you. I made them in Google Slides using circles and arcs. That's it, circles and arcs.
See, you hear that, people? All you need to get 500 plus subscribers on YouTube are circles and arcs.
Yep. So,
anything else? Unnecessary. You don't even need a computer.
Oh yeah, so I just realized some people might not be familiar with the standard like chemical coloring system, so I'll go through this. So the gray ones, like the dark, blackish ones, those are carbon atoms. Then the blue ones are nitrogen, and the lighter gray, like white ones, are hydrogen. So all of our DNA, a lot of our organic chemistry comes from carbon, hydrogen, nitrogen, and oxygen. Those are the main 4 in organic chemistry, I would say. I think those are the 4 most common elements in the human body. It's something like, 65% oxygen, then hydrogen, carbon, and nitrogen. So these are very important elements. And basically, adenine, it's just kind of a simple molecule to build. So that's why we use it in our DNA. You don't want to have something that's overly complicated. Otherwise, you won't be able to gather enough of it or make enough of it to actually write your DNA code. So if you ever see a DNA sequence written out as A, T, C, and G, this is the A. So, yeah, that's adenine. Also, adenine is part of ATP, I believe, adenosine triphosphate. So
yeah,
so this is the carrier of energy in your cells. So your cells, through the process of cellular respiration, like they turn glucose, of course, into carbon dioxide and oxygen. But along the way, through the Krebs cycle, they make ATP. When they use up the ATP, it's like a single-use battery. They break off one of the phosphate groups and make it a DP, which is adenosine diphosphate. And that needs to get charged again, essentially, with another phosphate. So it's this never-ending cycle.
Do you remember what the formula is for adenine, Max?
Adenine or adenosine triphosphate?
Adenine.
Let's see. Okay, we've got 5 carbons here, 5 nitrogens, and 5 hydrogens. So it's C5N5H5.
It's actually C5H5N5.
Oh wow! I should just
Close,
leave.
But no cigar.
Quit science now.
Okay.
Quit science.
Just quit the subject.
Oh my gosh. Yeah. Well, regardless, that is adenine. So adenosine triphosphate. It forms the base, and a phosphate group is essentially a phosphorus surrounded by 4 oxygen molecules. So imagine you have one of these adenines, and then you have a chain of 3 phosphate groups just tacked onto the end of it. And that is your little battery. And then to, transmit data through like proteins, like whatever your cell wants to do to move around, it just breaks off a bit of that. It just breaks off a phosphate and gives you adenosine diphosphate. So that's adenine. Okay, next we've got cytosine. So cytosine is the first one, I guess, in alphabetical order to actually incorporate an oxygen atom as well. So adenine is just pure, well, it's just carbon, nitrogen, and hydrogen. You don't even have your oxygen. But here, in red, we have our oxygen. So cytosine, I don't know much. I think adenine's kind of the only one that's useful outside of DNA. But again, as you can see, these are very simple molecules. This one is just a hexagonal shape with a few little, like, what do you call it? A few branches stemming off here and there. And it is simple, and it allows your DNA to, like, have a code. And it's pretty interesting because you have these 4 distinct chemicals so that each one is read differently when a protein is being created, but they also have to be simple so that they're easy to make. So they can't be so simple that they're indistinguishable, but they also need to be simple enough that they're easy to make. So yeah, cytosine is the C in the ATCG. Yeah, I don't know. There's not much to say.
What's your favorite letter of the alphabet, Max? While we're on that topic.
My favorite letter of the alphabet.
Yeah.
Favorite letter. I suppose it would be M. Yeah.
Mike, what would you say your favorite letter?
I got to go with M.
Yep.
The gang.
Yep.
I think I got to be an outlier here.
Are you gonna? Well, let me guess. Are you gonna go with S?
J.
J.
Yeah.
J.
Interesting.
Did you not know there was a J in my name?
What?
How have you
your
guys
middle
been
name?
spelling Sebastian?
Wait, what?
How have you guys been spelling Sebastian?
Well,
The
I've been
normal way that
spelling
any
it
normal person would spell
S-E-B.
Sebastian.
I've been spelling it Sebastian. Sebastian.
Same.
Yeah.
You missed the J.
Where?
Mr. J?
There's a silent J in it.
Where? Between which
Isn't
letters?
your middle name like John or something?
No.
No. It's between which letters?
Here, I'll spell it. S.
Where are you spelling this?
S. I'm saying it.
Oh, okay.
I'm saying it, Max.
Okay, fine. Say it.
S.
Uh-huh.
E.
Uh-huh.
B.
Uh-huh.
You got that, right?
Yeah, I got that. S-E-B.
A.
Uh-huh.
S.
Yes.
T.
T.
I.
I.
A.
A.
N.
Okay.
There you go.
That's it? Where's the J?
Where's the J?
The J is silent.
Oh my god. Okay, that's cytosine. So now we've got guanine. Guanine, I guess you could say, is a little more complicated because you have both a hexagon and a pentagon.
It's complicated.
But it's still a simple atom, just carbon, nitrogen, hydrogen. Guanine. So these base pairs fit together. That's why they're called base pairs, because they specifically go together. DNA has a double helix structure, like a staircase. So you have 2 sides, and they need to fit together like puzzle pieces. I forget which ones do exactly. Was it adenine and guanine?
It was adenine and thymine and guanine and cytosine.
Okay, so guanine goes with cytosine from earlier. So when the guanine is hooked up to the phosphate backbone of the double helix, it will, like, not entirely bond, but there are some intermolecular forces where it'll, like, hydrogen bond to the cytosine. So they fit into place.
3 hydrogen bonds.
Yeah.
3.
Yeah, 3 of them. Cool. Yeah, you can see those on the
there's
right
2
side.
for the other.
Okay. Yeah, so guanine and cytosine fit together with 3 hydrogen bonds. Cool. But yeah, so it's interesting. They're like tiny magnets, so they fit together. Or tiny puzzle pieces, whatever metaphor you would prefer. But what it means is that...
Are you magnet gang or puzzle piece gang?
Yeah, magnet gang or puzzle piece
Write
gang.
it in chat.
Yeah, team puzzle piece or team magnet. But anyways, yeah, they're interesting. I guess it's kind of insurance so that if like one side messes up, some things, I don't know. It kind of helps stabilize things and keeps it in place because there's also RNA instead of DNA, RNA, which is just one half of a DNA. It's a single helix. And the reason our, entire genome is encoded like that is because it's not as stable as DNA. So having two helixes and having these base pairs fit together helps keep our DNA stable over time. So RNA is still used in the body, but it's only used to carry short bits of information, so to transmit a little bit of information, which makes sense. So it's like a messenger. So if one message ends up getting like messed up, like that's fine. It's not going to be that big of a problem. It's just one molecule. But if the source is messed up, that's where most problems arise, and we'll get to that a bit later. Okay, so next we have thymine.
Well, do you know how guanine got its name?
No. How did guanine get its name?
Can you take a guess? Mike, what's your guess?
Someone, I don't know.
No idea.
No idea.
Max, what's your guess?
Does it have to do with like guano or something?
Yes, it does. It's correct.
Wow.
That's where they first found it.
What is
It's kind of gross. It's bat feces, but also fertilizer. So.
I see.
Yes, they found it in them feces.
Mm-hmm.
The most sophisticated of types.
Also a source of nitric acid, right?
Yeah.
So yeah. So that's interesting that they found it there.
Interesting fact.
Interesting facts. Yeah, exactly. My second guess. My second guess wasn't good, and I'm glad I haven't said it. Should I say it?
What was your second?
My second guess was Guam. They found it in Guam. The U.S. territory of Guam. Regardless, thymine. Thymine's the last one. And I don't know, I think it's...
No, there's one more.
Oh, well, there's one more, but it's weird. The last one is just like... It's like thymine's little brother.
It is the oddball.
Yeah.
It is the black sheep.
Yeah, we'll get to him in a moment. So thymine... I don't know. I would say thymine's the most like colorful or like aesthetically pleasing out of all of these. It just looks nicer. I don't know. I like it. But thymine fits with adenine. So they bond together in the double helix structure. There's not much about thymine, I don't think. Yeah, it's just, it is.
Yeah, well, this is the prelude to genetics. You gotta understand these chemicals.
Yeah, you just have to understand
before you get into the fun stuff.
Yeah, these chemicals.
This is just the cast list.
Exactly.
At the start.
Exactly. It's like talking about 0, 1, 2, and 3. It's like how much do you have to say about those numbers, right? Anyways, so
let's take a poll. Out of 0, 1, 2, and 3, what's your favorite number? Let's start with Mike.
Gotta go with number 1.
Number 1. Interesting. Max?
I would say 2.
2.
Yeah.
I gotta go 3.
Wow.
So you guys
really
like coming in
fields
second,
here.
third.
Wow.
I actually just like being the highest.
Oh, okay.
So Uracyl.
Yeah, Uracyl.
Well, hold on. You can be the highest if you're in first. I am in first place in the race. I am number 1.
The thing is, though, look at this, right? Look at Uracyl. So, if we compare Uracyl to thymine, like I'm going to switch back and forth quickly. Like, look at that. It's so similar. So Uracyl, pretty much, it replaces a single bond to a group of carbon, and that carbon is bonded to some hydrogens that thymine has, and it replaces it with just a single bond to a hydrogen. So it gets rid of a CH3 group and just gives you an H, which is a bit simpler, but it's only used in RNA. So what happens is, in DNA, you have your normal base pairs. You have A, C, G, and T. But when it gets transcribed into RNA, instead of a T, they replace it with a U, which is your uracil. I don't know why they do this. I don't know why RNA does it like this, but I mean, it's not that big of a change, but it's important because the way that a gene is used to make a protein is that you have one side of base pairs copied, but they aren't actually copied, they are transcribed. So the one they match with is copied. So if you have a series that goes adenine, cytosine, guanine, the result you'll get is uracil, guanine, cytosine, because you have to get the match of each base pair instead of the base pair itself. So uracil also matches with adenine. So adenine's like a 2-timing varmint. Right. So adenine will fit with both uracil and thymine. So when it's inside like DNA in the nucleus of a cell, it is like snug with the thymine. But when RNA is created, when a RNA message is created, they use uracil instead. So yeah, uracil is an interesting one, but it's just like a replacement.
All right.
Yeah.
Let's get into the main topic.
All right.
Or what genetics is all about.
Yeah.
Let's talk about genes.
Genes? Why are we talking about the pants company? Ha 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 ha.
I was talking about Gene Simmons.
Oh, okay.
Popular band Kiss, or Gene.
From A Catcher in the Rye.
Yeah, from A Catcher in the Rye.
And various other. Yeah. No.
Wow, Gene is not a common name. Wow.
Yeah. Whatever jokes, the typical jokes that they always have about genes, because it's a word that sounds like another word.
Gene Wilder. Willy Wonka.
Ah. First Willy Wonka or second Willy Wonka? That's a very important distinction.
Oh, he's the first one. The other one is Johnny Depp.
Oh, okay. Yeah, anyways, so we've got genes. Genes are... So a single base pair really isn't enough to really do anything, you know? I mean, it's like a single binary digit. It's like a byte or a bit. No, it's a bit. It's like a bit, right? So instead of having one bit, a gene is like an entire code, so an entire program. So you've got your zeros and ones strung out. You have multiple bits, multiple bytes, and that completes a code that can be executed. So say the code is executed when you hit the like settings app on your phone, right? Your phone is specifically waiting for the signal of pressure on that point of the touchscreen so that it can launch the sequence of programming that is the animation to open the settings app. And in many ways, our genes are really doing the exact same thing. Computer analogies are very useful when talking about genetics. I feel like genetics, it's so close to just computing. Like, we are biological computers.
Yes.
So you could probably make a computer that runs on DNA. Like, I wouldn't be surprised. Right?
So, Max.
Like,
yeah.
Anyways.
During our on our trivia episode, I asked you a question. I don't remember how many points it was, but I want to see if you remembered the answer.
Oh no. Okay.
Okay. What percent? And I'll give you a range of being 1% higher or lower. What percent of our DNA do we share with chimpanzees?
Okay, it's like 98%.
Right on the money. It is 98%.
Did I get that right last time?
I think you got that wrong last time.
Okay. Well, I know it now. So.
There you go.
All right. So genes. Let's get started. First, we have a series of base pairs. So this actually fits with what I was saying earlier, so I probably should have changed slides earlier. However, this is what we're saying. If you see, you can see each base pair matching up. You see the AT and the CG. You see the 2 hydrogen bonds with the H, or the A and T, and you see the 3 hydrogen bonds with the C and G up at the top. So, yeah, so this is the essence of what a gene is. It's just a string of these base pair characters that allow you to build a protein. So that's how genes function. That's how they create, that's how they affect things. All they do is create proteins. A gene can't say make you hotter, raise your body temperature, right? It can't raise your body temperature and just be this gene means raise the body temperature. It doesn't work like that. What would have to happen is there's a specific gene to release a protein that when it interacts with the neurons in the brain, it tells the brain to trigger heating within the body, right? So that is what a gene can do. Proteins, in the classic sense, when you hear protein, you usually think get buff, protein, muscles, but those aren't the only uses of proteins. Proteins aren't only used in muscles. Proteins are also used to carry information throughout your body. They are used as enzymes to speed up chemical reactions in your body, like digestion. So proteins can be a myriad of things, not just things that help you move. So genes, you can think of them as programs that create proteins. And so this is interesting because when is a gene triggered? How often does it create a certain protein? That gets into epigenetics, which will be a bit later. But it's a very interesting connection. So basically, a series of base pairs creates a protein. It's like a blueprint for a protein. So that's what you should imagine, right? Okay, so the first step in creating a protein is RNA transcription. So RNA transcription occurs when whatever sequence of events triggers your gene, the RNA polymerase comes in and it pulls apart the double helix temporarily so that it can build a little RNA sequence off of one side of your genes, one side of your DNA, right? So you start with your TACT, whatever, and as I said, it becomes the inverse, so it's your pair, your match. So the RNA actually says AUG, AU, whatever. So your RNA is presenting the flip side of what it read in your DNA. And so now that RNA transcript can be brought from the nucleus of the cell where the DNA resides, and that RNA, the little strip of RNA, can be brought into the ribosomes, I believe it's called, right?
Yeah.
Okay, so the ribosome is an organelle, just like your nucleus. So it's a part of a cell. And it is what produces all of the proteins that your cells produce. So the RNA is carried to the ribosome so that it can create a protein. And so the process of creating a protein goes from transcription to translation. So let's go to this. So this is protein synthesis, and this is your translation step. So you have your RNA, and now what happens is your RNA hooks up to amino acids in your ribosome. So the way it does this is that your body has 20 amino acids, and each one is attached to a little, I forgot what they were called. It's just a little molecule in your ribosome, and that molecule has 3 base pairs on it. Do you know what it's called?
What it's
A codon?
To Uracyl?
Yeah, kind of like a codon. I don't know what the official name for the molecules.
I think it is codon.
Oh, sure. So it's a codon.
I'm pretty sure that's what it is.
Okay. So basically, since we have 20 amino acids, that requires a minimum of 3 base pairs to describe every amino acid, because if you only used 1 base pair per amino acid, you could only get 4. If you only used 2, that would only get you 16, which isn't quite enough. So we need 3, which gives you 64 possible options of, if you have 4 numbers to pick from, right, and you have 3 slots, that gives you 64 possible combinations. So, in this way, each codon, each series of 3 base pairs, is your byte in the computer analogy. So each base pair is a bit, and 3 of them form a byte. So you have 64 possibilities, and you only have 20 amino acids. So some amino acids, I think, have up to 6 different codons. So some of them only have 1, but some have different amounts. So it's not a one-to-one correspondence. But every codon has 1 distinct amino acid that it connects to. So once your RNA is brought into the ribosome, each codon will fit just randomly. The codons connected to amino acids that are floating around the ribosome will fit to their matches, to their appropriate piece of RNA. And what you'll end up getting is a string of amino acids that forms a protein. So that's your whole process. A protein does need to fold together after it is created, but that is more of a factor of how the amino acids are shaped and what's their charge, their polarity, what kind of tails they have. So your protein will fold into the proper shape, and then bam, you have a protein, and this protein can go on to do anything in the body. Say insulin, it helps your cells take in glucose, or pepsin, I think, helps you with digestion. Then, there's a lot of proteins. So this is how. So that's how they're made. How is it made? Like that show. How is it made?
Yeah.
This is how proteins are made. There's one thing I forgot to mention with the RNA transcription, right? And that is that there is also something known as a stop codon. So, along with your 20 amino acids, there is also a 21st option that a codon can code for, and it's not an amino acid. It's literally just a stop sign. It just tells the RNA polymerase, this is the end of the gene. You don't need to keep going. If you were to keep going, it would just be excessive. This is all we need. This is the complete set. So there's a marker for where to start, and that's epigenetic. But the marker for where to end is actually within the base pairs. So it actually says, stop, you've gone far enough, right? And this is where some mutations can come into play, but that's a little later. Some mutations will mess things up using the stop codon. Others will mess things up by putting the wrong amino acid in. So we'll see that eventually. So, okay. Now we have epigenetics.
Hold on.
Yeah.
1 second.
Yep.
Whoever can get closer to this wins one verified Sebastian dollar.
Ooh, a verified Sebastian dollar.
So, whoever is closer to the amount of times that your DNA could stretch from the Earth to the Sun and back wins that dollar. So, Max, how many times can all the DNA in your body stretch from the Earth to the Sun and back? So 2 AU.
I will say 7. So 14 AU.
14 AU. Okay.
Let's go with one higher, 8.
Michael is correct.
Oh no.
Am I?
Well, no, you're not correct, but you're correct. You're
close.
closer. It's 600.
Oh.
What? Oh
okay.
That's crazy.
Well,
There you go.
My strategy worked
$1
again.
Yep. You get one verified Sebastian dollar.
We should
Yeah,
design
how am I gonna use it?
We should design and mint it.
It has the same buying power as a Zimbabwean dollar. So good luck.
Yeah, here we'll send you the design so you can print it out and take a picture with it, and then we'll put it on the Gravdamax Instagram. Yeah. That's what we'll do. Okay.
How much is a Zimbabwe dollar worth?
Oh,
that's what I'm checking right now.
You don't want to look it up.
$1 Zimbabwe is, oh my god. It's not even worth like a cent. It's worth less.
Yeah. They had a inflation.
This is really unfortunate.
They got rid of their currency because it was.
They just straight up got rid of it.
They switched to USD.
Oh my gosh, that's crazy.
Wow.
So they just straight up said, "Sorry if you have any money, it just doesn't count anymore."
I'm sure they like.
Oh, they switched to the euro. Either.
Oh, euro. Boo. They switched to USD, but they switched to euro. A lot of places actually do use USD instead of local currency. It's interesting. But
more tourist, maybe.
Yeah.
Mm.
Yeah. But before it was worth 1/5 of a penny.
I'm pretty sure. I'm not 100%, but I think the Philippines might use USD because the Philippines.
I think they do.
The Philippines was a Spanish colony, and then it became an American colony, and then it became independent. So it might have just held on to the U.S. dollar, just because it's easier than making a new currency. But
Philippines currency is the Philippine peso.
Oh, okay. Huh. I thought
which is 2 cents.
Okay. Well, anyways, so epigenetics. So you have your genetic sequence, right? But epigenetics describes things outside of your genetic sequence. So each cell in your body contains your complete genome. But that doesn't entirely make sense because if you think of the function of a skin cell, which is to just kind of like sit there, be somewhat stretchy so that you can move, not break, like keep germs out, compared to the function of a neuron, which is, everything that makes up your conscious mind, right? Those functions are very different, but they are both coming from the same exact set of instructions. So the question is, why don't your neurons act just like your skin cells, and why don't your skin cells act just like your neurons? And the answer is epigenetics. So epi, I think, is one of those suffixes that stands for outside of. I don't really know. I can't keep track of suffixes anymore. We have like 20. Like we have.
Doesn't it mean on top of?
Maybe. Who knows? Who knows?
Let me check. Yeah, upon or above.
There you go. So it's upon or above your genetic code. So these come in the form of, say, little markers on your genes that say, hey, hey, RNA polymerase, you should start a gene or start a protein here, right? Then they also come in the form of when you want to, like, say, in your neurons or your skin cells, they also come in the form of having your DNA that you don't want used, having it wrapped around, like, these little proteins so that they just cannot be used. So we'll see that in a moment.
Indeed.
Yeah. So first we have dominant versus recessive. So this is an interesting one. Every gene is either a dominant or recessive gene, I would say, right? Would that be a fair statement? Okay. So
yes.
Your genes, your genome is actually formed of 2 copies of every chromosome. So each chromosome in your body, that's why there are 23 pairs instead of saying 46, because they aren't 46, like they're not coding for unique things, a pair. A pair may not be identical, like they're likely not identical at all. However, they are still coding for the same thing, even if they are not identical. So, for instance, right, one genome might have a gene that codes for eye color, right? And on its paired genome, it'll still have a gene coding for eye color. It might not be the same gene is the problem. It just might be a different one. So that's where your dominant and recessive come in, because some common dominant versus recessive traits you might hear of are like eye color. I think that's the prototypical one. Then sometimes hair color you might hear, but it's typically always like they talk about eye color. So brown eyes come from a gene that is coding for melanin in your iris. And blue eyes come from a gene coding for no melanin in your iris. So what ends up happening is if you have one copy of the allele, as they're called, instead of types, they need a fancier word than type, so they call it an allele. So if you have one brown eye allele and one blue eye allele, the fact is that even if you just have one protein producing melanin for your eyes are still going to end up with melanin in them. Because, if you have two copies of it, your eyes will fill up with melanin, but if you only have one copy, they still will, because there's still there's still a sequence present to code for that protein, right? But if you have two copies of the recessive allele for blue eyes, then there's nothing present to actually produce that melanin for your eyes. So that's essentially what happens. It's hard to determine which genes will become dominant and which will be recessive sometimes. Sometimes it's not as simple as like what I described. Sometimes it's a really complicated process. But for genes that are just coding for a protein, right? The dominant gene will be the one that is coding for the functioning protein, and the recessive one is the one that's coding for no protein. So if you have at least one copy of that dominant gene, then even though the other copy may not be producing any functional protein, the other copy is. So it makes up for it. It's like having two kidneys, Yeah. It's like a fallback, fail-safe type thing. So yeah, that's dominant and recessive. Any comments or?
So I don't know if you mentioned this, but that little beam or the P thing that we have, that's called a Punnett square. And it's basically just a representation of how different things work. Like if the lowercase y's are recessive traits and the dominant y's, or the uppercase y's, the capital Y's are dominant traits. So they show how if one person with a dominant and a recessive mingles with someone with two recessives, their offspring could be any one of those boxes. And you can see which one is more likely. But here you see that it's like 50-50 in between one dominant, one recessive, or two recessives. But this also tells you that it would be impossible for there to be two dominants.
No, it's possible. Wait, say impossible.
Just not for
it's
this
impossible
particular.
On the left. On the right is what's called a trihybrid, and which is adapted dihybrid cross.
Yeah.
It's basically just a Punnett square on steroids. It's like, because this is how genes, it's a more accurate representation of how genes are, but genes and like your DNA, they're like a decahydroxy.
Yeah.
They're like prehensile, like cross,
Yeah. Basically, what this reflects.
Such a large number.
What this reflects, the trihybrid Punnett square is a series of 3 different genes, each with 2 alleles, a dominant and a recessive. So what this describes is that sometimes genes kind of work together to contribute to a trait. Because in school they always talk about either brown or blue eyes for the two, like as an example for dominant versus recessive. But the thing is, not everyone has blue or brown eyes. Some have green or like hazel or any weird color, right? There are so many different eye colors. So the question is, if there was just one gene describing eye color, then why don't we all just have one or the other eye color, right? So
Exactly.
genes are much more complicated than that. We have, like somewhere around 1 billion base pairs, right, in our body. So like in our genome. So that's just so much information to code. And I'm pretty sure everyone, like if you ever see in a movie when they do like a high tech, like an iris scan, you ever see that when they do that? When in like spy movies sometimes too, they'll like look down at the scanner with their eye and it'll be like, oh, you're allowed in because you are whoever. So everyone's eye is essentially unique, like a fingerprint. So there are so many different genetic factors that determine what your eye color is, what patterns appear in your iris, because an iris isn't just a homogeneous color. So what this trihybrid Punnett square represents is that there are genes that interact with each other to contribute to a trait, and different combinations will result in different expressions, so you need to see the probability of each combination and just how it works. So yeah. Cool. I'll go to the next slide. Gene expression. So gene expression here is basically asking you, when will your genes produce a protein, right? Because we went through the process of protein synthesis a little earlier. And you might be questioning, if a gene is just a blueprint for a protein, then is every gene just producing, say, like 1 protein per minute or per hour? Like, is there just a set rate? Does it just keep on going and going? Well, the answer is no, because that wouldn't work. Sometimes you need a protein more than other times. For instance, insulin, I think it's just before you have, like before you eat, when you're starting to get hungry, insulin increases in your blood because your body is expecting an intake of glucose soon, and that is actually part of what contributes to hunger. The increase in insulin in your blood tells your brain, okay, we got to eat something, we're hungry, it's time for more glucose. So what this means is that when the cellular environment is experiencing a relatively lower level of glucose, that triggers the production of insulin. So that means that there's some factor on the outside of the DNA that is scanning for glucose. And when it has an appropriate level of glucose, it says, okay, RNA polymerase, do not come here. We're good, we have glucose, you are not needed. But when the glucose levels start to taper off, the glucose leaves, so the signal is then turned off, and the RNA polymerase comes in and produces insulin. Things can also go the other way. For instance, the lactose or lactase, whichever one's the enzyme.
It would be ACE.
ACE, okay.
With ACE is an enzyme.
Yeah, so lactase is used to digest lactose, not with a T at the end. These words are like tongue twisters, really, all the scientific words. But yeah, so the lactase enzyme, the sequence of DNA that will produce the lactase enzyme, is usually turned off because you don't need to digest lactose all the time. You don't need to digest milk all the time. So what will end up happening is once your body starts to detect lactose present in your cells, so that means that you've just like drinking milk or had yogurt or something like that, then it triggers the signal and says, okay, we need lactase now, so RNA polymerase, come here so we can do that. So it's interesting. It really is, because if you think of every reaction your body has to different stimuli, like when you get too cold, if your body was producing a protein 24/7 that like told your brain that you're too cold, that wouldn't work, then you would just constantly try to heat yourself up until you got too hot, and you would still think you're cold all the time. So your body needs to be adaptive. Your body needs to be like, okay, how hot is it, right? What's the temperature like? And it needs to have triggers on the DNA so that once it gets too hot or too cold, different genes will start being, or different proteins will start being produced to change your behavior and maintain homeostasis, right? That's the end to every paragraph, I think, in bio class. Like 50% of the
100%, yeah.
Yeah, like half of every like test question or like worksheet answer, it's always just to maintain homeostasis. That is your catch-all like useful phrase in bio classes. It's like the species or like the organism does this, this, and this, and it does it to maintain homeostasis. But
Yep.
yeah, so that's that. Should I go to the next slide?
Yeah.
Okay. So now we have gene inactivation, and this is what I was talking about earlier with your neurons acting differently from your skin cells. So, one way that your body does this is during cell differentiation. So, this happens throughout your life, but it also happens a lot like when you are, when you go from a single cell to a full baby, right? I think it happens when there's around 100 cells in your body. So we all start as just a homogeneous, pluripotent stem cell. And the word pluripotent means that it can become every cell. So this one cell can become every cell in your body, and eventually it does. So once you get 100 of these pluripotent stem cells, they start to differentiate. The ones on the outside become ectoderms, the one in the middle become endoderms, and then the ones between the outside and the middle become the mesoderms. It's kind of like layered like that. So the way that cells differentiate is by permanently deactivating some genes. So say that, because as I was saying earlier with gene expression, right, your genes will have little switches on the outside that react to outside stimuli. So if it detects glucose, it'll produce insulin. But you don't want every cell in your body to produce insulin after it detects glucose. Some cells should just be able to detect glucose and not do anything, because you only need certain cells to produce that insulin. Same thing with lactase. Yeah, lactase. You only need the cells in your stomach and digestive system to produce lactase when they detect lactose. You don't want every cell in your body to start producing lactase when it detects lactose, right? So some genes need to be permanently turned off to differentiate your cells. And the way that they do that, I'm pretty sure, this is one way I know of, there might be more, is that they actually have these larger proteins that they wrap the DNA around. And they just wrap it around this protein, then they grab another one and they wrap it around there. And this keeps the DNA tied up tight so that it cannot be used, it is inaccessible, so it is essentially turned off. So in your brain, the neurons have turned off some of the genes that your skin cells have kept on. And that is all due to the environment that these cells were experiencing while you were developing from like a single cell. The cells that detected, okay, I'm on the outside of the cell cluster, deactivate different genes than the ones that say, okay, I'm in the middle of the cell cluster. So it's interesting because while every cell does have a full genome in it, they're not using all of it just because they don't need to. So, yeah.
Yeah.
Cool.
Is it time for Hereditary?
Yeah,
Heredity?
sure.
Sure. Okay, sure.
Michael
Heredity.
can't speak part
I
37.
can't speak properly.
Heredity.
We
I
go
would
to the
also-
main man himself.
Yeah.
Sorry.
Go.
Correct.
Go
Gregor.
ahead.
Greg.
Gregor.
Gregor.
So heredity is when a gene passes from parent to offspring. And as we were talking about earlier with dominant and recessive, those rules partially determine how a gene is passed on, but the whole concept is called heredity. So, like you always
Let's
hear.
get to Gregor. Let's get to Gregor.
I'm
Let's
just gonna
get to
say
Gregor
one more thing.
himself.
Let's
I'm
go.
just gonna say one more thing. You always hear, like, if you have a condition, like your doctor might say it's hereditary, or they might say it's not hereditary. And when it's hereditary, that means that it is something that your parents had, or that they carried, so that your grandparents had, or something like that. So yeah. Anyways, okay. Gregor.
Gregor Mendel.
Oh God, we're gonna get copyright struck.
Gregor.
0.5 seconds of NFL music.
Gregor
Let's
Mendel.
go, Gregor.
Now it's
So
my time to
Gregor
shine.
Mendel. Oh, you got it.
So Gregor Mendel, he is a scientist who discovered how traits are passed through generations using peas, green and yellow peas to be exact. All right, and he discovered, all right, that throughout his 7 years of testing, that yellow peas were dominant to green peas, and realized that hey, there's a dominant and recessive traits here, right? So every time he I forget what he exactly was looking. I think it was length, color, and amount of peas in the pod, maybe. That was his whole goal, and he realized that each gene, like, kind of mixes and passes on through each offspring. So yes, that is the basics of Gregor. Denied at his university level because he's sad. Thank you.
Wow.
There you go. Yeah. So, Gregor Mendel, father of genetics, actual Chad, and we have two beautiful illustrations of him in
Thank
the
you.
top left and top right from our very own Jam Has Been.
It's
And Gregor Mendel he established 3 laws. They're called Mendel's 3 laws. And just briefly, there's the law of dominance, and a dominant trait is a trait whose appearance will always be seen in offspring. In other words, dominance describes the relationship between 2 alleles. If an individual inherits 2 different alleles from each of its parents, and the phenotype of only one allele is visible in the offspring, then that allele is said to be dominant. Secondly is the law of segregation. So a parent may have 2 distinct alleles for a certain gene, each on one copy of a given chromosome, and this law states that these 2 alleles will be separated from each other during meiosis.
Okay.
And thirdly is the law of independent assortment. And this states that the way an allele pair gets segregated into 2 daughter cells during the second division of meiosis has no effect on how any other allele pair gets segregated. So in other words, the traits inherited through one gene will be inherited independently of the traits inherited through any other gene, because the genes reside on different chromosomes that are independently assorted into daughter cells during meiosis.
So he's basically noticing that like your genes like reshuffle somewhat randomly during meiosis.
yeah.
Yeah. So Greg figured all this out. Definitely a Chad. And great guy. Overall great guy.
You know what's really sad though?
What?
At the time he discovered it, no one believed him. It took, I think, 30 years later, he died, for everyone to realize it, and they awarded his dead body with a prize of some sort. I don't know.
Wow. So,
high prize.
Question: did he discover all this before DNA was discovered?
I think so.
I could figure that out for you.
Let me.
I'll find out when DNA was
Okay.
or not invented. Discovered.
Invented. When was DNA invented? About
When
3.8
did
million
Mendel
years
discover
ago.
genetics?
Yeah.
Oh, great. It's not going to tell. Oh, okay.
When did
1865 is his results were published.
Oh, so that's
late 1860s for DNA.
oh.
Ooh.
Huh.
So he did it like right before.
So that's fascinating because.
So he's a legend.
Yeah, absolutely. It's interesting. A lot of
I would. Yeah,
a lot of scientists, they are able to do work and produce laws without fully understanding a concept. You know, they can do it before their time. You know, before something's fully understood. Like, I think it was Kepler came before Newton, but Kepler came up with the laws of planetary motion without even knowing about gravity. Like, without Newton's laws of gravity. He was just like,
of a savage.
yeah, exactly. He's just like, they all move in ellipses, and the amount of time it takes them to move through the ellipse is determined by the area between whatever, like the slice. I can't word it correctly, but you know what I'm talking about.
Yeah.
And then also the one that they move slowly if they're farther out. He discovered all that before Newton was like, oh, okay, this has got to be gravity. So he made his laws of planetary motion before gravity was discovered. And so in a similar way, Gregor Mendel was able to make these laws of heredity without even knowing about DNA. Yeah.
Yeah.
That's cool.
Very cool.
Yeah.
Yeah. Gregor may or may not be my real dad. He actually, we could figure it out through genes.
Yeah. So Gregor Mendel, when did he die? In like the 1890s or something?
I think so. He
died in 1884.
So that means that Sebastian is at least 136 years old. Am I getting that correct?
You figured me out.
Quick, get him. Figure his secret out.
Get him. Well, so you are at least...
How have I survived this long?
Yeah, how have you survived
Maybe
this
it's
long?
talent. Maybe it's Maybelline.
His father told him the secret to
my
genes.
Gosh. Yeah.
Exactly. I just grabbed all the genes that allow me. I'm like literally half tort because I can live so long.
Okay.
But
You're
anyways,
like
let's hop over to the next slide with
Next slide.
one of the best artistic pieces of this century.
Okay.
Cool.
Hidden. Detected. Yeah. Anyways, that's fun. Okay. I'll go to the next slide.
Thanks, Gregory.
So, we have dominant diseases according to Gregor's Gregor's law of dominance. So, these genes, if you just have one copy of it, it will present in you. So that is like brown eyes, or I think brown hair, I think, as well. Yeah, so you've got a lot of dominant traits. But sometimes a dominant trait can be harmful. And this is an interesting situation, okay? Because a dominant genetic disease, it can't kill you before you have had offspring of your own. Otherwise, that dominant disease will wipe itself out. Because say there's a disease and it has a dominant allele, and like it kills people like at a very young age, right? Then that gene will never spread, and so the disease would be eradicated, which is great, you know. But what happens is there can still be dominant diseases. It's just that they don't affect you until you're in like your 40s or 50s. So you've already had children. You've already spread this disease to another generation. But it can still be fairly malicious, like, and malignant, even afterwards. Have you heard of like chronic familial insomnia. It's very rare. Have you heard of that? I'm not sure I'm getting all the words right. I know it's insomnia, like familial, chronic familial. What it does is it's a dominant trait, and it produces these proteins that like slowly attack and destroy your brain, but it takes decades for it to actually become damaging. So for decades of your life, you are just living normally, like you don't know that anything's wrong. And then what ends up happening is that you start to get insomnia. And that insomnia develops, and usually by the time that people go to their doctor, I mean, there's nothing that can really be done because it's a disease that's been affecting them their whole lives. It's just that the symptoms haven't shown until they're like in their 40s. So then they go and they're like, I can't sleep, like what's this? And it takes a while because it's so rare, but then eventually they figure out that it's this disease, and then it progresses to like early onset Alzheimer's, things like that. So it's still pretty like harsh disease considering the human lifespan like should be like it's in like 78, I think, depending on if you're looking at developed regions or undeveloped regions. I think in the US it's 78. So the lifespan is typically 78. And this disease can kill you like 30 years earlier. But most people have already had kids by then if they are going to have kids. So the result is that this like malicious disease can spread even though it's deadly, which is just crazy.
Yeah. So some dominant diseases are Huntington's disease, neurofibromatosis, neurofibromatosis type 2.
Okay. There's 2 types.
Yes. And polycystic kidney disease.
Okay. Yeah, so these are all chronic. They take a long time to fully develop.
Yeah.
So that's the problem with dominant diseases like this. You don't know until it's too late.
What is Huntington?
That you have it. So
it turns you into a hunt.
Yeah.
Your nerve cells break down in your brain.
That's okay. That's not.
And
a
it
hunter.
typically starts in your 30s or 40s.
Oh.
So that sucks.
Yeah.
Yeah.
Yeah. So it's the typical trend. It's always in your 30s or 40s. But yeah. So dominant diseases, they're a problem because even if you have a healthy copy of the gene, it's not enough. It's that like it's kind of like if the brown eyes, blue eyes thing was reversed, right? Like you have a healthy copy of the gene to make your eyes brown, but the blue-eyed gene, not only does it just like not produce the protein to make your eyes brown, it also like attacks that protein and damages it, so that it will force its way into being a dominant trait, and it's just crazy like that.
We all have blue eyes here, right?
Yeah, I think so.
I think so, yeah.
Alpha Squad.
We're all UTs.
Yeah.
UT blues.
We also all have brown hair, right?
Mine's kind of black.
Yeah. Well, brown to black.
Yeah, mine's brown. Yeah.
Yeah. Interesting. So, yeah. So dominant diseases, they will affect you when you're older, but not when you're younger, which is just weird. But it makes sense because any dominant disease that springs up, that's like, hey, I'm a new genetic disease, like, what's up? And it kills you very young, it's just going to disappear from the gene pool instantly. So the way that these dominant diseases remain is that they only affect older people. So the ones that only affect you once you get to a certain age are the ones that will remain, which is just craziness. Yeah, okay. So next we've got recessive diseases. And you see the red got more intense. That's because these diseases are like even worse,
Yeah, these are the bad boys.
Yeah, so recessive diseases, they will affect you when you're younger. But here's the thing, right? If you have one copy of a recessive disease, then you're fine. You'll live a normal life. You will have no ill effects. There's like absolutely nothing wrong. In most cases, some cases it can be like very minor, but really like you're fine, right? But if you have two copies of a recessive disease, then you will not be fine because you will have that recessive disease. And these recessive diseases can be nasty. They are, I'm not gonna, I wouldn't necessarily say worse because it depends on what you define as worse. But instead of waiting until you're in your 30s or 40s to affect you, recessive diseases will often affect people that are very young, because the recessive disease does not have the same constraint that the dominant disease has. Because in a dominant disease, everyone that carries the gene also carries the disease. So if they die very young, then the gene will not spread. However, a recessive disease can have people that carry the gene but don't actually have the disease. So it means that when someone has two copies of it, it can be extremely deadly and kill them in their childhood. So that's where like a lot of horrible like diseases come from these recessive traits. Yeah. But then there's also this specific pedigree right here is actually a recessive recessive, what's the other one? I think sex linked, yeah. Right? I don't think
X-linked.
Yeah, so
it's autosomal and something else.
Well, this one's not autosomal. I think it's just called sex linked.
Yeah.
So
and X-linked.
Yeah, so this is really getting
autosomal
our
and X-linked. Those are the two.
Oh, okay. So this is getting our what do you call it? This is getting our COPA rating. This is a sex-linked recessive disease, as in it is linked to your 23rd chromosome. It's linked to the X chromosome. And what happens here is, since men only carry one copy of the X chromosome, whereas women have two copies of an X chromosome, a recessive disease that is situated on the X chromosome will affect every male that carries it, because they only have one copy, and if their copy is the disease, then that's it. But for women, if they have one healthy allele of the disease, then they can carry the disease but not have it. So that creates an interesting situation because it means that the children of someone that has this disease will also likely carry it. But it depends on if it's male or female. So I think it's like if someone's mother has, so if a woman carries this disease, actually has the disease, that means they have two copies of the allele. So that means that all of their sons will also have the disease because they are getting their X chromosome from their mother. And their daughters will be carriers, assuming the father doesn't have it. So that's interesting. A disease that follows this pattern is color blindness. So it means that men are much more likely to be colorblind than women. And that's
my dad's colorblind.
Oh, really? So
Gregor Mendel.
Oh god.
No, my actual dad.
Yeah, it's a 50% to 25% chance male to female.
Yeah,
that's kind of busted.
Yeah, definitely. So what happens is, since men, it's like all or nothing with your one allele. It's like if your one allele has the color blindness gene, then as a male you will have color blindness. But if you are a woman, then it takes two copies of the genes to get color blindness. So that's why women aren't as likely to be colorblind as men. Yeah. But yeah, it's interesting though, because even though your father, Sebastian, has color blindness, that has no bearing on whether or not you will have color blindness, because your X chromosome is from your mother and your Y chromosome is from your father. And since since color blindness would be on your father's X chromosome, you are not a carrier, or like you are not affected with it. But that does mean that your sister is probably a carrier, I think, right? Yeah.
Yeah, that's how it works.
Yeah, so one of her X chromosomes from your mother, the other from your father.
Yeah, I'm not colorblind.
What?
Yeah, because your father
you're
gave
not
you the
colorblind. Yeah, you wouldn't be colorblind if your father is. But what it does mean is that your sister is a carrier of the colorblindness gene. So she's obviously not colorblind, I think, right?
Yes.
Okay, so she's not colorblind, but it means that her kids could be. So, like, there's a chance.
Some other recessive diseases include cystic fibrosis, sickle cell anemia, and like a lot of others. A lot of diseases are recessive.
Yeah. And yeah, this is what I'm talking about of the recessive diseases. Obviously not color blindness, but some of these recessive diseases can be really bad. Cystic fibrosis, like it messes with your lungs, makes you prone to infection. So those have really short like prognoses. Like they can kill people quickly. But let's see, what else? Oh yeah, you were talking about sickle cell anemia. So that's an interesting one because some diseases aren't like 100% diseases, because sickle cell anemia, people that only carry one copy of the gene, they don't have as bad like sickling of their cells. It still can happen, but only after they've like put themselves under severe stress. But if they only have one copy of the gene, then they are more resistant to malaria, which is interesting. Because malaria, it affects the red blood cells. It's, bloodborne disease, like carried by mosquitoes. So you can actually, like some genes are like a blessing and a curse. So if you're just a carrier of the sickle cell anemia trait and you don't actually have the full disease, then you will be resistant to the malaria disease. So that's an interesting way. Like our genetics are so interconnected. So it's hard. Like even when we talk about genetic editing and stuff, we don't really have that as topics in this video because it's expansive enough just talking about genes like operations as usual. It's hard to get into the like genetic editing, but it's difficult to do genetic editing because we don't know the full scope of what one edit will do. Like you go in just for like a cosmetic purposes. Say someone wants to like have a different eye color, right? You go in and you change their eye color, and like, oops, you just like created like X-Men. What you gonna do about it, right? Like you go to like give them a different eye color, and then now they're like starting to move metal with their mind, and they're like, oh, what have I done? Right? So our genes are crazy interconnected. So yeah, that's it for me. I'm gonna go to the next slide. Any other comments?
The next slide.
Nope. Cool. I think we're good.
Okay.
But speaking of X-Men,
mutations. Yeah, mutations. So there are a few types of mutations that occur, and we're going to go through them. But on a basic level, a mutation is when your cells are copying either through mitosis or meiosis, and your cells, like the copying mechanism, makes an imperfect copy of your DNA. So this is a problem regardless of like when it happens. It's just always a problem. Sometimes it can be corrected. I wouldn't say sometimes. I'd say most of the time. I mean, if you think about it, we have trillions of cells, right? And most of them work completely fine. Like maybe one in every like million base pairs or something like that will have an error, but then most of those are corrected later, right? It's only like, it's just crazy how well oiled a machine our bodies are. But the problem is, it can take just one mutated cell to cause like cancer. So it's crazy. Our cells have to be perfect in their operations. And they mostly are. Like by any standards, if you were to look at like a manufacturing plant and compare it to how our cells operate, you would be like, wow, our cells are just so much better. But there's just so many of them that over time, the probability makes it so that one of them somewhere will mess up, right?
Yeah.
So a mutation that occurs early in development, so like during meiosis or during the first few like mitosis steps, like when you're starting as a single cell, those can result in like diseases that affect your whole body, like from day one. Like that's where I believe new genes come into play. Blue eyes was a mutation like 10,000 years ago, so that must have occurred during the meiosis or the early stages of mitosis in some person, and then they just like had blue eyes, and then everyone's like, huh, interesting. Um, so that's like how that works. But mutations can also occur just, in a fully like formed like an adult human, just because your cells are constantly undergoing mitosis, because cells die and new cells need to be made to replace them. And when a mutation occurs there, that's where you see cancer come into play. Because a cell decides, I am not going to die, like I have an error. Most of the time when a cell is produced that has an error, it knows, and it's like, okay, it kills itself pretty much. It's called apoptosis. So a cell will undergo apoptosis if it knows that it has an error. It's like, okay, I don't want to mess everything up, so I'm just going to apoptosis. And if that doesn't work, then most of the time your immune system can pick up the baton or whatever and get rid of those cells. But when a cell manages to have a mutation in the apoptosis gene, so it won't like go through apoptosis. It also has a mutation in some gene that allows it to seem like a healthy cell, but it's really not a healthy cell. And it has a mutation in the reproduction, like the rate of mitosis. With those three mutations, that is what will produce a cancerous tumor, because it can stay hidden from the immune system, and it will divide, and it will not know when to die. So yeah, so that's interesting.
Yeah.
One of my favorite concepts,
Let's head into them.
Oh yeah, I just want to say one of my favorite concepts though is something called a hypertumor, which is where a cancer
your
cell.
cancer gets cancer.
Yeah, your cancer cell mutates again and decides to rebel against the original cancer. So someone's cancer can get cancer. I think it was a news story. Their cancer got cancer and killed their cancer, so then they were fine. Which is like, literally, that's like fighting fire with fire or something like that, Like that's crazy that can happen. But yeah, anyways, so mutations. So we start with nonsense.
If you think that any salad dressing besides Hidden Valley, the original ranch dressing, is the best, you be speaking nonsense, which is exactly what this mutation. Ranch dressing is God's gift to humanity. It is Icar. It is the absolute blood of reality itself. Without ranch dressing, do you seriously think U.S. economy would be as booming as it is? In 2008, in the Great Depression even, Hidden Valley Ranch wasn't being used. Economy, gone. 33% of Americans unemployed. 2008, boom, bam, gone. No one can afford a house. Why? Hidden Valley Original Ranch wasn't being used. Now business is booming, right? Everything is all tied together based on the combination of whatever the hell they make ranch dressing. What do they make ranch?
I'm looking at the picture. It looks like milk, of some form. Tomatoes, lettuce.
Maybe I should do
and
that on
peppers.
stream. I make my own ranch.
Oh, we should do a stream where we just make ranch, just cooking show.
The combination of mayonnaise, sour cream, buttermilk, salt, black pepper, garlic, onion, chives, parsley, and finally dill come together to create the best dressing the world has ever seen.
It's interesting, actually.
You can get off the stream.
Wait, what did he say?
Ranch is a bad dressing.
You
Any
must
dressing.
have a nonsense mutation.
I hate
You're speaking gibberish.
dressings.
You're speaking gibberish.
No. How can you even eat a salad without dressing? That's just like
I don't eat
leaves.
salads. I'm too skinny
okay.
for one.
What? That doesn't make sense. That's
Yes, it
more
does.
nonsense.
See, he's speaking nonsense. He's not making any sense. He's
You're
speaking
too
nonsense.
skinny for salads. What does that even mean?
I don't like salad.
Okay. Well,
Anyway.
What I do like, though, is pasta.
okay.
Wow. I see you've adopted a healthy lifestyle.
I really like pasta.
Look, I think your
I
opinions,
really like pasta.
your opinions on
I really
salad,
like chicken as well.
you just need to find someone that can make good salad. That's really it. Because I agree, salads can be garbage, but like
salad is just a bunch of leaves in a.
I know, but like that's if you get your salad from well, I don't want to say any brand name with a negative connotation, but a fast food restaurant. If you get your salad from a fast food restaurant, like it's okay to talk about ranch because we're talking about it positively, so it's not like they're going to sue us. But if I were to talk about how absolutely horrible the salads are at this fast food restaurant, they might not be so pleased. But yeah.
This fast food restaurant we're talking.
Mm-hmm.
Who makes a salad?
I think they all make salads, all the fast food restaurants, just in a vain attempt of look, we're not entirely unhealthy, but the salads are just horrible. You need good salad, like homemade.
Oh yeah, we can't really talk about them in a bad light.
Yeah.
Because Wendy's restaurants see our stream, they might not be so pleased.
Yeah.
And especially Wendy's restaurants see that we're talking about it in a negative light, they might press charges.
Yeah.
And Wendy's restaurants take us to court. It won't be good.
Yeah. Yeah, I don't think these restaurants will be loving it when we talk negatively about them on our live stream. Yeah. But anyways, okay. So a nonsense mutation. What occurs is, as you can see, you have your normal sequence, right? And you have this codon TGC. I think it should actually be UGC. I wrote the U up. I messed that up. But just pretend this is a normal sequence, right? So you have TGC right there. And in the mutation, only one thing is wrong. The C is an A, so you have TGA. And so you might think oh, that's just one thing wrong. How bad could that be? It can be horrible, okay?
Now you have bones coming out of your fingers, okay?
Exactly.
You have extra bones
you're
everywhere.
Wolverine.
You are Wolverine.
So TGC codes for, I don't know which, it codes for a amino acid. So it just produces another amino acid, and it tells the RNA polymerase to keep going to complete the protein. There should be a ... here. We're just looking at a snapshot, right? But TGA is actually a stop codon. So this tells the RNA polymerase, okay, we're done, but they weren't actually done. It's like saying halfway through that you're done, right? You can't do that. That won't work. So you only have the first half of a protein being produced ever, and that will not work at all. Say this is the insulin gene, right? That would result in non-functional insulin. So then you would need external sources of insulin, which would give you. I think that's. I don't think it's specifically a nonsense mutation, but that would be type 1 diabetes because your body cannot produce insulin because it doesn't have the correct instructions to do so. So TGA, this stop codon, it's kind of like getting very unlucky, right? Because a base pair mutation can cause a variety of things, but if it just so happens that this base pair like replacement gives you a stop codon, then it like cuts your protein in half. Like you only get the first part, you don't even get the second part. That would be like watching, I don't know, the first part of, I don't know, give me a movie that comes in 2 parts. That would be like watching the first part of It and then never seeing the second part, right? Like chapter 1 of It and then just not chapter 2. You would be like, oh my gosh, what am I going to do, right? Yeah, it just does not function well. Yeah. So, there you go. Anything else you got? That's it for me.
You don't like ranch.
Okay.
I don't know, man. I'm not a fan of ranch. I mean, I'm not a fan of any topping, actually, except cheese.
Cheese.
Let's eat cheese.
Okay.
Cheese and actually, I like that's pasta.
Pasta.
I'm not
Is
a
it
fan
all
of ketchup.
pasta?
Not a fan of mustard. I'm not a fan of most of these things.
Do you like honey mustard? That's the real question.
No,
Honey mustard is kind of trash.
Oh, are you serious? Did I just hear
Dude,
that?
what am I a fan of?
Dijon mustard is so.
Okay, honey mustard is good. I also like normal mustard, but honey mustard is also good, okay? It's good.
I'm telling you, Dijon mustard, bro. So
What?
superior.
Is that just regular mustard?
It's the spicy. Like, I'm talking about
Oh, yeah.
the spice, like, gold spicy.
Yeah, I think I've had
mustard is
that.
my
No, I like that
favorite
too.
brand.
I like that too. I'm just saying, I
Or
also...
Gildan's spicy brown mustard.
I do like that too. I don't like honey mustard at the exclusion of other mustards. I just think it's good. It's like sweet. It's good.
I can't agree with you.
Okay, whatever. We'll just continue. Next we have deletion.
With delete. Delete.
Yep. Delete.
Delete. So
Hope you
that's
get the
from Death
reference.
Note. This
Yeah.
is the guy that deletes things.
He deletes people.
I am a delete. Your
So
Twitter.com.
quite simply, you can look at the normal DNA, then you look at the deletion, and
Mhm.
oh boy, there was a deletion.
There's an A that's gone.
No, there's
Not okay.
there's actually a second C in the second group, the GCC. The C is missing, right?
Yeah. Yeah.
But the problem is, your DNA doesn't really know when a codon stops, you know? It's just like going in groups of 3, and it's just saying once it gets to the polymerase, and once it gets to the ribosome, it'll become groups of 3. But while it's in the DNA, it's not like there's commas separating them, you know? It's not like there's nothing to indicate where a group of 3
I would like to
starts.
address chat. Oh, okay.
Chat going off?
No. Well, we have a chat.
Okay. We have Ryan Rudin. We have ranch is underrated unless it's on pizza. I'm a monster.
You know what?
You are a monster.
Okay, let's dissect this. Ranch, yes.
Uh-huh.
Absolutely underrated. It deserves to be the president of the United States of America. Unless it's on pizza. I'm not sure whether he's saying that it's good on pizza or it's bad on pizza because of how the sentence is worded.
Oh,
But
yeah.
I'm guessing that he's saying it's good on pizza. And you know what? Have at it. If that's how you want to use ranch dressing, like there's even stuff salad pizza. So maybe he's putting it on his salad
No, wait, I
pizza.
bet he's saying ranch is underrated unless it's on pizza, which would mean that it's underrated in every situation other than the pizza one in which it's trash,
pizza.
right?
Yes, so if he's saying it that way,
Yeah.
sure, yeah, ranch on pizza, little strange. But you know what? It's a free country.
Yep.
And ranch will soon become the new USD. And
we're just going to be trading bottles of ranch instead of...
Republic.
We'll all be trading new currency.
He changed it! He said, correction over. Ryan, I was with you. I was with you to the grave. And then you stab me in the back and change it to overrated?
I agree.
Okay.
Leave! No! Leave!
Hey, he likes it on pizza at least, so...
I can't believe it.
We agree on
I cannot believe it. You turn on me like this? X2, Ryan? Oh. And he's like, as only if it's on pizza is it good. You need to reshape your taste buds. Your taste buds must have had a deletion mutation because there's something missing.
All right. Anyways,
and I agree with him. Ranch is overrated.
Anyways,
you're testing my limits.
So
Boo-hoo.
You have deletion. What happens here, right? The second C in the GCC is not present. And your DNA, it doesn't have anything to indicate where a group of 3 starts and where a group of 3 ends. So the result is that everything is shifted off one. And it messes up everything, like, so bad. It's like taking a sledgehammer to the DNA code. Because now, right, instead of GCC, you have GCT. So that could just code for one other, like, wrong amino acid, right? Which is bad enough as it is. Like, a lot of mutations are caused by just one wrong amino acid. But on top of that, the next part, which is supposed to be TGC, is now GCA, which is just, again, a completely different amino acid. And then your AAA just becomes AA, and then whatever would be next. Again, this is just
AAA. Oh.
Just a snapshot of a gene. But yeah, a deletion mutation causes so much problems just because it messes up the framework. Because everything's neatly packed 3 after 3 after 3. And then you just have a 2 and fill in from the end of the next 3, and it just messes things up horribly. So, yeah. So
thanks, deletion.
What?
Really doing a good job today.
Thanks, dude.
Yeah, so deletion.
There you go.
Yeah.
I wish Ryan Rudin would get deleted.
Oh my god.
Wow,
Anyways.
that's very violent. Should I delete you? Let me delete your voice.
The war of the ranch versus non-ranch users.
I don't know. That's going to be my next video.
That's what you're going to start.
Ranch dressing is the best dressing.
I just don't see.
You should do history of ranch dressing in 120.
Yeah, okay, but Mike, this is coming from the guy that literally only subsides off of pasta.
This is coming also from the guy. Like, I'm talking about me right now, who only needs pasta and chicken. All right.
Oh my gosh.
I get my energy.
So it's chicken Alfredo, and
you just sound so malnourished.
I don't like them together.
You don't like, so the 2 things you eat are chicken.
I mean, I've had it
pasta.
before, but it's just not cool.
Exclusively separate.
Yeah, I don't like mixing my foods. What can I say?
It's like the diet of a Minecraft character.
It's funny too because I weigh. I'm not like anorexic or skinny. Mm-hmm. Well, I'm skinny, but not anymore.
Mm-hmm.
I have a stomach.
Okay. We all do.
Mm-hmm.
Thanks, Mike.
Anyways.
Mike is just slowly obtaining organs.
He's slowly obtaining more organs. He is 75%. He's still missing a lung.
By the second I grow.
He's still missing a lung, the 3rd ventricle of his heart. Yeah, no. Anyways, yeah. I love this GIF right here.
The 3rd ventricle of his heart.
Yeah. Oh, there's
my heart is very hard work.
Well, aren't there the upper 2 and the lower 2? Or maybe the upper 2 go by a different name.
Yeah, there are 4 chambers.
Oh, the 4 chambers.
But it's the top 2 are atrias.
Okay.
Or atriums.
Yeah. Sorry, he's missing the third chamber is what I meant.
Or just atria, rather. That's the plural. Just atria.
Okay. Sorry, I meant chamber.
Yeah, that's what we're doing in anatomy.
Okay.
Every fiber being with like.
It was like what? Yeah.
You what?
Yeah, exactly.
But anyways, let's talk about insertion.
Yeah. Okay. Call back.
Much like this railroad spike,
Mm-hmm.
which I'd like to believe that Phineas Gage carried around for the rest of his life,
Mm-hmm.
was inserted into his skull and brain.
Mm-hmm.
Sometimes something can get inserted into a genetic code. So if you look at the insertion part, you can see that a third C was inserted.
Oh yeah, third C.
Right there.
Yeah, exactly.
Which throws everything off by one.
Yep. So now it's got the same problem as deletion, really, is that it just messes everything up, because now instead of TGC you have CTG, and then instead of AAA, CAA. So deletion and insertion are equally destructive. That's what I would say. They're equally destructive, but they are very distinct things. One is adding something and another is deleting something. But in the whole grand scheme of things, they are equally as damaging. They do pretty much the same thing.
They're both bad boys. No.
Yep. Also, you know how two wrongs...
Stay away from me.
You know how two wrongs make a right? Or they yes. Don't, but in this case, if you were to have an insertion mutation and then very shortly after a deletion mutation, it could keep things not as bad, because things would be off for a little while, but then they would return to normal. And it's possible that that wouldn't be as destructive, which is interesting. So in the case of DNA, two wrongs can make a right. So tell that to your parents when they say that two wrongs don't make it right. That's your response. What about insertion and deletion genetic mutations? Get wrecked. Anyways, so that's it. Anything else you guys have? Nope. Cool.
Nope, sir.
Then we got Missense.
Yeah.
He a little confused, but he got the spirit.
Yeah. So it happens here.
So this is just when one thing is transcribed improperly.
Yeah, or one thing is copied wrong. This isn't about the RNA. It's about your DNA source code. So as you can see, instead of TGC, we have GGC. So for the TGC codon, a different amino acid is in place with the GGC codon. So it can be very damaging, but compared to just the absolute catastrophe that you see with nonsense or deletion or insertion, missense is comparatively mild, but comparatively mild when it comes to genetic errors is not really a good place to be, Mm-hmm. So missense can still be really bad. It replaces one amino acid with another. And remember what I was saying about proteins needing to fold correctly. So one half of your protein will fold correctly, the other half will fold correctly, but right in the middle, you have this missense mutation, and it could be on the spot of a critical hinge, and because it's using a different amino acid, it does not bend properly, and now you have a useless protein. So, yeah. So that's the problem. Or it could just be a partially useless protein, maybe one half still works and it gets the job done, but that's the aspect where I'm saying it's not as damaging as a deletion, insertion, or nonsense mutation. But it can still be pretty bad. I'm trying to think of some diseases that are caused by this. I think... Oh, well, I know one. FOP. Have you heard of FOP?
Stoneman syndrome?
Yeah. So it's just one missense mutation, so one wrong amino acid. And what this disease does is whenever you get a bruise, you bump your hip on a table or something, instead of healing with more muscle tissue, your body will heal with more bone tissue. So parts of your body that's supposed to be muscle will become bone over time. And that's obviously bad. Again, very rare. So, like, don't worry about it. Sometimes when you talk about very rare and interesting diseases, I always feel like, oh my gosh, like, is this going to be me? You know, do you ever feel like that? Like when you're talking about a disease?
Am I the one to go next?
Yeah, exactly. I die. Like you start talking about, say, the coronavirus for a bit, and then you feel your throat like hurting a little bit, and you're like, oh no. You know?
I have the virus.
Yeah, like the paranoia a little bit. But yeah, so don't worry about that. But that missense mutation can be pretty bad. So yeah, okay. So that's it for me. Anything else you guys have?
No, I don't think so.
Cool. Okay.
Nah.
Finally, we have silent mutations. So this is, I don't know how, but you used the wrong formula and got the correct answer. You know, I actually did this once on a test. My teacher did not give me the points. Yeah, I know. It was on a math test. I did the right formula. I did like something stupid. I think she gave me partial points. It was that I was just, you know, like, this was in pre-calc. So I was just doing some algebra, reshuffling, whatever, like, you have to move everything to the different sides. And instead of subtracting something so that you would end up with, like, say I had y equals x, for instance, but it was more complicated than that, right? I had y equals x, and instead of turning that into y minus x equals 0, I turned that into y divided by x, like something like that. Regardless, whatever, like, I messed it up. But I still got the right numerical solution. So I was like, when I got the test back, I was like, huh. And my teacher was like, yeah, that's interesting, right? And I'm like, that sure is interesting.
Yeah, the closest I've ever come to that is I guessed what an answer was.
Yeah.
You know, like, I don't think I've ever done it on a test, but I've done it on homework where I'll be, like, in a rush to do the math homework, and I'll just, like, I did this more, like, in earlier years, because now you have to, like, actually do your just to make sure that you have it for the test.
Yeah,
exactly. Teachers don't check it or not.
Yeah.
But in, like, 8th grade, teachers would check it, even though it's stupid. The concept of it is stupid. But I just wrote some random answers down, and, like, one of them turned out to actually be the correct answer.
Wow.
And it was insane.
Yeah. But yeah, so that's in essence what a silent mutation is. It's when you use the wrong formula, but you got the correct answer. So let's take a look here, right? In our third codon, no wait, not in our third codon. In our second codon, where we're supposed to have GCC, we have GCT. But remember, there are only 20 amino acids and 64 possible combinations of codons. That means that some amino acids have multiple codons that code for them. So GCC and GCT just so happen to code for the same amino acid. So if you have a mutation where the second C in your GCC becomes a T, you're fine. Like, you don't need to worry about it. Your protein is not going to be messed up at all because your ribosomes will still read like GCC, GCT, I don't care. Either way, I'm going to put this same amino acid in. And then your protein, you know, it'll fold, and it'll fold the exact same way because there's nothing different about the GCT amino acid or the GCC amino acid. They are identical. So a silent mutation is technically a mutation, but literally nothing comes of it. Nothing bad happens. So that's the used the wrong formula and got the right answer. So that's really fascinating. You know, that's just luck, really. Essentially, the nonsense, missense, and silent mutations are all the same thing. It's just one wrong letter. But nonsense results in a stop codon, missense results in a different amino acid, and the silent results in no problem. So that's pretty cool.
Yes, sir.
Yeah, because there might be two versions of a gene that produce an identical protein. Like, and you might have that. Like say your eye color gene. Your eye color gene might have a mutation in it, but you'd never know. No one would ever know because it's no problem, because it's just a silent mutation. So, yeah.
Nice.
Yeah. Okay, so that's it. Let's see. Final thoughts.
Final thought.
Genetics. They're crazy. So much can go wrong,
Yeah.
But amazingly, it's like it goes right, most of the time, or at least some higher percentage of the time.
Yeah.
There's a lot of like horrific stuff, especially when you get into like radiation.
Mm-hmm. Oh yeah, radiation.
You get, like whack mutations.
Radiation is not
also
good.
With like inbreeding and all that, you get terrible mutations.
Mm-hmm.
But it's interesting that genes can actually turn out correctly when there's like actually trillions of possibilities.
Yeah.
It's kind of nuts that they actually can form functioning humans, and like 7 billion, almost 8 billion functioning humans at that.
Mm-hmm. Yeah, definitely. Genetics are really interesting. And I did realize as we made this episode that we didn't really talk about, the technology and the way that we might be able to edit genes in the future. But I think that would need to be a separate episode because we are already at an hour 50. So that wouldn't have really fit. So that would need to be a separate episode at some point in the future. But genetics are really interesting. I mean, it's so similar to computing, I think. I don't know why. I just love the computing analogy here. It just works so well. Like most analogies, you have to stretch a bit, but there are so many parts of the process that you can see as being identical, like the little flags on your genes, your epigenome that says when lactose is present, produce lactase. That's like hitting your spacebar on your computer to change slides. So it's really fascinating how much of a parallel there is.
Yeah. What you thinking, Mike?
Genes.
Thanks, Mike.
Yeah, thanks,
Contributing to the Gravity Max live stream as always.
Yep. Yeah, hit it, Mike. You remember that?
I may have just dropped something off my desk.
That was fun. The live sound studio. Anyways,
Boom.
You can watch that episode if you want some laughs. The aliens episode. It wasn't very informative, but it sure was fun. But yeah, so thank you for being here, Michael. You've been a great guest, as always. You can come back anytime you want. Yep.
It is always a pleasure being here.
Thank you. Yeah, we had you last season for, life extension technologies. So, yeah, always good to have you here. And I would also like to thank my co-host, Sebastian, for being here as always.
Of course.
Ooh.
Yep. Yeah, great co-host. Always, fun as always. Great Sunday tradition. Most people, take a family walk every Sunday. Instead, I do a Gravity Max live stream every Sunday. Yeah, so
that's a funny joke.
Talk about science every Sunday. So that's fun. So that's it for me. I will end the stream unless you guys have any more comments.
I don't got anything.
Cool. All right.
Pretty good stream.
Yep.
Thank you, everyone, for watching.
Yeah, thank you for watching.
See you next Sunday.
Yeah, see you next Sunday.